Hello everyone, welcome to the next episode of my science podcast. Today I'm absolutely delighted to welcome Professor Dianne Newman, who is a molecular microbiologist and a professor in the Division of Biology and Biological Engineering and the Division of Geological and Planetary Sciences at Caltech. Her research focuses on the bioenergetics and cell biology of metabolically diverse, genetically-tractable bacteria, as well as biofilm survival physiology in the context of human chronic infections and crop rhizospheres. She was awarded the National Academy of Sciences Award in Molecular Biology for her discovery of microbial mechanisms underlying geological processes. So thank you so much for joining me today. It's an absolute pleasure to have you on my podcast.
It's a pleasure to be here, Martha.
So to start off with, may you tell us about your early work and how you progressed to where you are today?
Certainly. So I am a microbiologist, and one of the most extraordinary things about microbes is their metabolism. They can eat and breathe things that humans can't even dream of. And so if you're someone who appreciates microbial metabolism, this gives you an incredible array of problems that your interest can contribute to studying. And when I began my career, I was very, very fortunate to be hired at Caltech to launch what they called at the time their program in molecular geobiology. And this was an idea to capture all of the sequence information that was at the time becoming ever more easy to acquire and ever more inexpensive to acquire, and to leverage this revolution in molecular biology and sequencing information in a way that could teach us something about the history of the Earth, and in particular about the history of life on the planet. And if one wants to understand the early history of life, you need to work on microbes, because they've been around the Earth for at least 3.5 billion years. And during that time, their metabolic activity has completely transformed our planet. It's thanks to ancient cyanobacteria evolving oxygenic photosynthesis that today we are able to respire the oxygen that's in our atmosphere. But before oxygen was around for us to respire, there were all sorts of other things, weirder things like rocks. And so I was very curious, how is it that a microbe breathes a rock? And these were some of the questions that were ones that, piqued my curiosity when I began. And as I can tell you in a little while, are questions whose answer has a theme that extends well beyond the context of microbe-mineral interactions. And I can elaborate on that in a little bit.
Wonderful, amazing. So with your earlier work, what are the basics of microbe-mineral interactions, including how bacteria catalyze mineral formation, transformation, and dissolution?
Well, as I said just a little while ago, the beauty of microbes is their metabolic diversity. And as I said, they're able to eat and breathe all sorts of things. And when I say eat and breathe, that's sort of putting it into human-centric terms. Of course, microbes aren't really eating and breathing. They don't have digestive systems or circulatory systems, respiratory systems. They're much, much simpler at the cellular level. Yet what they have that is phenomenal are their electron transport chains. And when you understand how these electron transport chains work in some detail, what quickly becomes apparent is that they're very modular. And so you can have many different types of electron donors or sources of energy, if you will, into these chains, that when the electrons are passed from one component of these chains to another, this enables energy conservation. And so it's the mixing and matching of what is the electron donor and what is the electron acceptor that enables energy conservation, that at the end of the day is what underpins studies of microbe-mineral interactions. And so a mineral can either be a substrate that can donate electrons to a system, or it can be a substrate that receives electrons. So whereas, for us, you just had dinner, I suppose not too long ago, you probably had some tasty food that was full of organic carbon that you were able to combust by respiration. And in so doing, you were able to extract the electrons from your food and dump them onto oxygen. That's, at the end of the day, what's going on in our mitochondria, which are nothing more than ancient bacteria. But bacteria can do these type of energy conservation combustion reactions with many, many, many different things, including minerals. So my lab studied how it is, what are the particular processes, what are the specific enzymes that cells need in order to catalyze more exotic metabolisms, to effectively respire something that is poorly soluble, that cannot, like oxygen, diffuse into the cell, but is outside. So how do you breathe a rock, if you will? These are the type of things that we're trying to figure out early on.
Amazing. So how do these processes relate to cellular energy conservation? Well, you just said how they relate to energy conservation, but how do they relate to membrane organization as well? And how these affect the geochemistry of their environment?
Yes, so it's all about understanding, the details of how the electrons are moving around. So it is the oxidation and reduction reactions that really sustain all of life. That's what powers energy conservation for any living thing. It's a redox cycle of some type. It's really quite phenomenal and quite elegant and beautiful at the level of electron transport and bioenergetics. So what we have to do, if you're trying to understand how electrons move around to weirder things like insoluble minerals, is use tools of genetics, biochemistry, cell biology to learn what are the necessary players along that pathway. What are the enzymes and where are they in the cell? And so many of the bacteria that I've studied over the course of my career are what you call gram-negative bacteria, which is an old-fashioned term that basically means that bacteria have two membranes. They have an outer membrane and they have an inner membrane. Not all bacteria are structured like that. Others have different types of cell walls. But for these gram-negative bacteria, the membrane, which is the inner membrane, is the one where all of the magic of energy conservation takes place in. And so if you are a bacterium and you're respiring a rock, you then have to ask, how is it that this rock outside the cell allows electrons to somehow pass through that outer membrane and then ultimately work their way into the inner membrane in order to either donate or accept electrons from the electron transport chain, which is localized in the inner membrane. So that's generally the type of question, and then it's really the answer to that question, once you know the mechanistic details of how it works, that can then provide much broader insight in terms of how in nature microbes are doing these type of things. And I'll be able to give you a more detailed example in just a moment, but that's sort of the starting answer.
Wonderful. So why did you then stray away from geobiological the geobiological stream of work?
Well, it was really a very natural progression, and in some ways I feel like I have never left the geobiological perspective behind. It's still very much part of what I do. What led us to shift our focus in some way was the realization that one of the major problems that we were working on, which was how does a microbe respire a rock that's outside the cell? The answer to this came for the organism we were studying in recognizing that this organism produced beautiful redox active metabolites, small molecules that could be excreted from the cell, that could go and react with the rock at a distance, be chemically transformed, and then be recycled, be taken back up by the cell, and in so doing catalyze a continuous stream of oxidation reduction reactions, both within a cell and extracellular, outside. And the more we thought about this, actually we realized this quite early on. My very first graduate student, Mario Hernandez, who was my first student from Argentina. She was in the environmental science and engineering program at Caltech. She and I realized that the cell didn't really know what was oxidizing these molecules at a distance. It just cared that something was oxidizing them and it was getting them back. And so whether it was a rock that was serving as the oxidant, or whether it was molecular oxygen, from the cell's perspective, it didn't really know and it didn't really care. And so we started thinking about what are the type of organisms that produce these redox active metabolites. And what we realized is that there was a very important one named Pseudomonas aeruginosa that is very famous, not in the micromineral field, but it's very famous more in the infectious disease field because it's a major pathogen of individuals who have chronic infections, chronic wound infections, or individuals with the disease cystic fibrosis. This organism has an ability to thrive in the context of infection. And so we started getting curious whether this ability to conserve energy that was enabled by the shuttling of these redox active metabolites, whether that might be a very generic type of metabolism that could be understandable as something that would support microbes when they were living in conditions where there were many different cells of their type around them that were all competing to respire oxygen, leaving in the mass of such cells, which we call a biofilm, cells in the interior that were starved for oxygen. And thinking that under these conditions, being able to produce your own electron shuttle at high cell density that could somehow go outside of the cell and react at a distance with an extracellular oxidant, whether it was a mineral or whether it was oxygen, it didn't really matter, might be a very broad strategy that sustained many different forms of cells when they were growing under conditions of being in a biofilm. So this became a very general hypothesis that we sought to test. And it turns out that that idea has really held up. And indeed, we now have actually quite a bit of evidence that this form of metabolism is very broadly relevant.
It's brilliant that you can start off in one direction and then it takes you in a complete, well, not completely different, but it goes in a different direction that you weren't imagining to start off with. And I think that's part of the beauty of science, really, the unexpected.
Absolutely, you said that very beautifully. I totally agree with that.
Yeah. So my next question is, well, you found that in response to phosphorus limitation, when it's not bioavailable, bacteria can produce phenezines in situ to liberate phosphorus in the form of phosphate from mineral surfaces. What implications does this have on other works, such as new fertilizers that are being designed or made?
Yeah, so this is actually a direct fallout of the work that we had done showing that these redox active metabolites, you just gave them a name, phenezines. So phenezines are my favorite example of this broader class of compounds that I have been calling redox active metabolites, that when they go out of the cell, if there's a mineral in the environment, and this mineral is something like an iron oxide, these minerals are very well known to bind inorganic phosphorus. So there's an attraction, an electrochemical attraction, a charge attraction rather, that the phosphate will stick to the positively charged mineral surface. So when the phenezine, this redox active metabolite, goes out, it is able to react with the iron mineral and donate an electron to the iron mineral, which chemically it's a reduction. It reduces the iron, it converts it from ferric iron to ferrous iron. And when that happens, it changes the mineralogy in such a way that the phosphorus can be released. It doesn't bind as tightly anymore. And so this is a way that organisms have of helping solubilize phosphorus. And interestingly, these phenezines and other molecules like them are regulated by a couple of things at least. We know for sure they're regulated by high cell density. So as I was saying earlier, it's when you have a big biofilm that, a cell might need to make another electron acceptor or electron carrying molecule. But in addition, they're known to be regulated by low phosphorus. And so you get sort of two bangs for your buck. You get intracellularly the ability of these molecules to preserve intracellular redox homeostasis and energy conservation, while at the same time, when they go out and they're recycled extracellularly, they liberate phosphorus that is adsorbed onto these ferric iron minerals. And so the potential relevance of this is thinking in an agricultural setting where we want to be able to better utilize fertilizer in a more efficient way. How might it be that we could think of making use of the property of organisms that produce these type of metabolites to make phosphorus more bioavailable? Could it be that if we were to seed certain soils with organisms that had the ability to make these phenazines, that they might be able for the community in situ, under conditions that are really relevant in nature, to be able to allow phosphorus to be solubilized. This is something that is an interesting idea, and we're just beginning to work towards thinking about how we might test that hypothesis.
Yeah.
And one of the cool things is that we know that phenazine producers are found in soils, and particularly in rhizospheres, which are the domain of the soil in the vicinity of plant roots. There's very good evidence from all of our genomic repositories that organisms that have the ability to synthesize these phenazines are quite ubiquitous. We find them in just about every rhizosphere that has been sampled so far. So how can we harness that going forward with synthetic biology? We're gonna try to find out.
It would be a real possibility then. Yeah, amazing. So my next question is, what did you find were the defenses that Pseudomonas have evolved against their own toxin, Piocyanin? And how does this change our view surrounding how antibiotic resistance occurs?
So something I have not yet called Piocyanin is a natural antibiotic. I've been referring to it deliberately as a redox active metabolite because that's really what it is. It's a redox active metabolite, and yet for many years people sort of thought of it exclusively as an antibiotic. And this is for very good reason. It's because its redox activity can generate, if there's oxygen in the environment, it can generate reactive oxygen species that are quite reactive and deleterious. And this has been shown for many cellular systems to cause toxicity, and it's very true. However, it's not the full story. These molecules, as I said, also play a role when oxygen is absent in helping the cells maintain redox homeostasis and conserve energy. They may also play roles in nutrient acquisition. So what their function is really is very much impacted by what the local microenvironmental chemistry is like. So in this regard, that my geobiology training has never left me. I always am thinking about what is the chemistry of the environment around these cells and how is that impacting what they're doing and what the molecules they're making are actually doing. So we recognized that these so-called natural antibiotics have the capacity to be both very good for the cells, but also toxic, not only to other cells, but to the cells that produce them. And so we started to look to understand, why is it that organisms that make these reactive metabolites aren't killed by their own toxic molecules? And we found there's a lot of good reasons for this. We and others in the field who've been studying these for many decades have now built an appreciation for the cellular defenses that are necessary to protect producing organisms from their own metabolites. And they include defenses against oxidative stress, ways of controlling the efflux of these molecules efficiently so that they're not accumulating in the cell and doing a lot of damage.
Mhm.
There are undoubtedly other mechanisms yet to be discovered of how within the cell they are localized in such a way as to not intercalate into DNA or react in a damaging fashion with iron sulfur containing proteins, for instance. There's still a lot more to learn, but we have done enough at this point to show us that these metabolites induce particular responses, and I'll give an example, efflux pumps. So these are membrane-bounded enzymes that are able to take a molecule and sometimes modify it, but the most important thing is just get it out of the cell fast.
Yeah.
And it turns out that it's this ability to pump them out quickly that helps the producing cells survive, in part, along with other functions. But what we were able to show recently is that even different bacteria that do not make these molecules themselves, but which have evolved in an ecological context like the soil alongside the producing cells, that they in turn have evolved the capacity to withstand them as well. And that when you have a producer side by side with another organism that doesn't make them, but has co-evolved with it, this other organism has the ability to recognize these molecules, turn on the same efflux pumps, and get rid of them. And the relevance this has for antibiotic treatment is that many infections are polymicrobial, and many infections, at least for people who have chronic bacterial infections,
Mhm.
many of these arise from soil bacteria that have a shared ecological and evolutionary history. And what's really important that's often forgotten is that the golden age of antibiotic discovery came from people like Sir Alexander Fleming and Selman Waxman in the United States, taking soil bacteria, realizing they made penicillin or streptomycin, natural products from soil bacteria or soil fungi, different microbes. And then synthetic biologists, synthetic chemists rather, evolved these molecules, designed them into now the standard antibiotics we use today in hospitals. And yet structurally, they resemble a lot of the natural antibiotics that have this, very long evolutionary history from coexistence of microbes and chemical warfare in the soil. And so it's very important to actually remember this shared history, because what it does is predict how today, when you give an antibiotic to a polymicrobial community, it may be that its efficacy is modulated by the type of natural metabolites that members of this community are making. And
Mm.
so it means that we have to think a lot harder about how we do antibiotic susceptibility assays so that we capture these metabolites that can be made in these communities, whether they're thriving in the soil or whether they're thriving, within a tissue in the human body that's infected. The chemical milieu of that environment is going to dictate how the organisms respond, and that in turn will dictate their sensitivity to the conventional antibiotics we use. So
Mm.
this is, I think, a really important application that we're pursuing.
Yeah. So possibly when testing antibiotics, it ought to be done in terms of high density conditions, co-culturing with other cultures as well. And so they've got the competition for the resources as well, so they can, the process will be more realistic and hopefully more effective in
Absolutely.
creating antibiotics.
Absolutely. Absolutely. You nailed it. No, what we need to do is be able to represent the actual conditions that organisms exist in the context of infection in the laboratory, so that we can do better experiments, so that we can get at mechanisms that are more directly relevant to how they're actually living. And we're getting better and better at doing this as a community, but a lot of the history of microbiology has been done sort of abstracted from the native context. And so I think this is something for you and the next generation of younger people going into this business to realize, is that there's an opportunity now with technologies having advanced so far that we're going to be able, there's no excuse for performing experiments on the laboratory bench that are divorced from fidelity to the actual environmental context. We can We can do both. We can do mechanistic work at the bench under environmentally relevant conditions. Yeah.
Amazing. So my next question is in another paper that I've read, in your work with Fleischmann. Is that how you pronounce it? I'm sorry if it's wrong. Yeah.
Yes, Professor Sarel Fleischmann or Fleischmann. I think he would be fine answering to either one. He's at the Weizmann Institute of Science.
Amazing. So with him, you altered an enzyme and produced it, well, was able to produce it in larger quantities, which was one of the issues that you started off with. And then in combination with an antibiotic, it helps the antibiotic kill the bacteria in the oxygenated outer regions of a biofilm in a way that had not been seen before, leading in short time to a significant reduction in the total number of viable biofilm cells. So what was this unusual way of killing the bacteria that hadn't been seen before?
Yeah, well, we're actually still in the very thick of working out the exact mechanism, and we have a couple of different hypotheses that we're going to test, which are different, but they're not mutually exclusive. I'll give you just one of them as an example. So the synergy that we saw when we added this novel enzyme that degrades one of my favorite phenazines, a phenazine called Piocyanin, was that when we gave biofilms this enzyme, and in addition gave it a clinical antibiotic commonly used called tobramycin, we saw synergistic killing. And one reason why that might be the case is that piocyanin, as I told you, it is toxic to the cells that produce it under certain conditions. And those conditions where it's most toxic are when they don't have very much carbon and they have a lot of oxygen. This actually is a condition where they have a difficult time managing their own toxic molecule, and it leads to lysis. Okay, so in these biofilms, in the oxic outer sphere of these biofilms, there's extracellular DNA that results from these cells being self-poisoned and some of them lysing. That DNA has been shown by other investigators in my community to be able to bind tobramycin, which is a cationic antibiotic. And so if you added an enzyme that Fleischmann helped us make in greater quantities much more readily, what this would do is diminish the amount of eDNA, lysis, that was being generated, allowing for greater penetration of tobramycin. So that could be a way that they're acting synergistically. That's one hypothesis. And as I said, there are other explanations that could work in a completely different way. So we're going to find out, hopefully in the next few years, which of it is the case.
Amazing. So you've touched on this a little bit already, but what are exactly extracellular electron shuttles, and how do these help bacteria at the bottom of a biofilm survive when they lack ready access to oxygen or other electron acceptors used to conserve energy?
Yeah, well, so this gets back to where I was telling you in the early part of our conversation how what my first graduate student I realized is a situation where cells would need to make these extracellular electron shuttles, which is yet another name for what I was calling redox active metabolites, is when they're growing in a film, biofilms. Okay, and so you can think of a biofilm as like a bacterial version of a tumor almost. And it's well known from cancer research that you need to have delivery of oxygen to sustain metabolism in tumors. And the way that works is something that many people, have studied for a long time. And so the delivery of oxygen, or rather the delivery of oxygen equivalents, and by that I mean a molecule that can receive electrons to allow for cells, within these biofilms to remain, metabolically viable, to conserve energy. This is something that these extracellular electron shuttles can achieve. And so whether you call them extracellular electron shuttles, redox active metabolites, or give them a specific name like phenezines.
Yeah.
That's what they're doing, is that they're serving like a molecular snorkel almost, allowing for cells in the interior of a big biofilm to pass electrons, in to the cell, but allow them to go all the way out and get recycled, these molecules, so that they can sustain the metabolism of the cells in the interior. When they're unable to get oxygen, because the cells on the outside are respiring that oxygen and consuming it faster than the oxygen can diffuse into the center. And so that's why they need another solution. They need to make their own electron acceptor, if you will. And that's what these shuttles are doing for them.
Thank you. That makes sense. So jumping back to the question beforehand, why does piocyanin, why does it bind to extracellular DNA from the cells that have died or been, with their own toxins and spilt their contents into the biofilm? We'll do that first and then we'll move on.
Yeah, it's because it's what's called an intercalator. So DNA, you've seen the beautiful structure of DNA. It's like a ladder, right? And between the base pairs, you can have certain molecules that slot in them, that they're planar, and they can just find a happy resting spot in between the base pairs of the DNA ladder. And these type of molecules are known as intercalators. So ethidium bromide is something maybe you've used when you've stained a DNA gel. It's a fluorescent metabolite that intercalates into DNA, similarly to how piocyanin does. It slots in between the base pairs, and it has a fairly nice affinity. And so when you're running an electrophoresis gel and you want to stain it, at least back in the old days, you would add ethidium bromide, and then you could see it under the right light box where the DNA bands were.
Amazing. Thank you very much. So my next question is, how has the transcriptome imaging method, named parallel sequential fluorescence in situ hybridization, aided your research on biofilms where growth conditions can change at a microscopic scale?
Yeah, well, this was a very exciting new method that a very talented former postdoc of mine named Daniel Dar, he's now, I'm proud to say, leading his own group at the Weizmann Institute of Science in Israel. He developed this method when he was in my lab in collaboration with a colleague, Long Kai, who's a professor at Caltech too. And what it allows is for us to look at hundreds of transcripts within individual cells found in these biofilm communities. And because of my interest in metabolism and realization that there are bound to be gradients in where oxygen, for example, can be found, we wanted to become better able to figure out what cells are actually doing in any specific place, to actually visually see what they were doing. And so Daniel Dar developed this powerful method to do spatial transcriptomics, where we could literally light up genes of interest at the level of single cells within these biofilms. And what it showed us, one of the many things that it showed us, is just how extraordinarily sensitive, exquisitely sensitive these biofilms are to their microenvironment, and that over very tiny distance scales, you can have microbes experiencing very different chemical realities, and then they adjust transcriptomically. And so I think it really reinforces what I was saying earlier, which is it's very important when you're thinking about microbes, or really any living system for that matter, to think about what the microenvironment around that living system is, because it shapes what it does. And so this is a tool that allows you to get a glimpse of what the cells are doing. And one of the next frontiers is to have better tools to actually measure and visualize what the chemical and physical microenvironment is also like.
I see. Amazing. So I guess, what are the implications of that in terms of antibiotic resistance? So imaging that and being able to advance the research that's going on in that area.
Yeah, well, we're just really at the very, very beginning of what I suspect is going to be, profound changes in our understanding of what microbes are doing. This is a method that literally allows us to see things we've never been able to see before. And so when you're charting a new world, whether it's at the microscopic level or if you're an explorer of, a continent back in the 15th century, when you see something for the first time, you begin to be able to navigate and to identify and see what's present in any specific space and if there's any patterning to it. And it's only after you begin to chart the landscape of something and see what the patterns are and where things are that you can develop hypotheses that get more into, why do you see these patterns? How is this working? And then it's only at that level, once you get to a more of a mechanistic understanding of why things are where they are, that you get the kind of insights necessary to control a system. And so for these, in the cases you asked, antibiotic approaches, what I hope is that from this methodology, we're going to be able to capture what organisms are doing really in situ within a real infected environment and learn what processes matter most. And that will give us then a hypothesis that we can use to guide future experiments to ask, okay, if we perturb this particular pathway that we see as important here, will that give us a new handle on our ability to control these organisms? That's the hope, but we're really at the early stages now because this is such a new method, and I can't wait to see what's going to come.
It's fascinating. So my next question is, how does a phenazine degrading agent comprised of a Piocyanin D-Methylase reduce antibiotic resistance of the bacteria? And what chemical processes does it interrupt in order to do this?
Yeah, well, this is something that we were discussing just a little while ago with that enzyme that my collaborator Cyril Fleischman helped us with. And it gets back to the recognition that phenazines are fulfilling all sorts of important roles in biofilms, and they're modifying the biofilm properties as they go. And that if you're trying to solve the problem of antibiotic tolerance, which is actually a different thing from antibiotic resistance per se. So tolerance is when you have a physiological state that is able to withstand antibiotics, as opposed to you have something that has evolved in the organism that allows it to grow really quickly in the presence of antibiotics because, for example, it can now pump it out better, or the target of that antibiotic has mutated in such a way that it's now invisible to the antibiotic. That's resistance. What is becoming clear is that tolerance and resistance are linked, and that if you're an organism that can be more tolerant of antibiotics, meaning you can be in a state where you withstand antibiotics, you're not growing, but you're just surviving. And then when antibiotics go away, then you can grow again. That ability to tolerate antibiotics allows for evolution to occur that leads to mutations that then confer actual resistance. So these things go hand in hand. It's quite an
So to finish off, what would you like to see happen in your area of research in the future in terms of what you feel is currently lacking or hindering the efficiency of the research process?
Well, what I would like more than anything else is for molecular and cellular biologists to really embrace the fact that life exists in context, and that the context is outside of our laboratory bench. The context is out in nature or in the context of, infections that we're trying to solve in the human body. And that if we really want to understand how life does its thing, regardless of what the context is, it compels us needing to characterize the actual context and take the time just to observe. I think that as much as I love molecular and cellular biology's emphasis on mechanistic research, that is something that really gets me going. I crave to understand how things work. I feel that that's very intellectually exciting to be able to do experiments where you seek an understanding of mechanism.
Definitely.
But what I think has gotten lost a little bit has been the recognition and the respect for the fact that before we can get into the detail of how something works, we need to understand what the landscape is to begin with. At some point in any scientific investigation, there's a phase of just observation and of discovery and of seeing what's, what's relevant. And so I think that because molecular and cellular biology is now an old and venerable field, and many of the problems are ones that, have a many decadal history behind them, that some of the state-of-the-art stuff has sort of gotten decoupled from the very early stages of discovery. And what I would like is for that early phase of characterization, observation, to be brought back into the fore and to help frame the way we go about mechanistic research. To realize that life only exists in context, and if we really want to understand how things happen, we need to get better at understanding what the real context is that motivates everything to begin with. So that's where I see an opportunity.
Wonderful.
And technologies are getting so good for this that your generation is really going to have some amazing things to discover.
Oh, definitely. The opportunities are so vast and so exciting. And it's all because of your amazing research right now. So thank you so much on behalf of everyone for what you're doing. It's wonderful. And today's been absolutely fascinating. I've learned so much. And I'm sure all the viewers have as well. So thank you so much for joining me today.
It's been a pleasure. Thank you. And I hope you go do research when you get to college. I think you've got a really bright future, and I hope you keep your curiosity and inspire many others your age. It's really quite wonderful.
Thank you. Thank you. So thank you everyone for listening, and I'll see you next time.